Good morning. Thank you very much for your kind introduction and for the invitation. Today, I will present my work at Ragon Institute in Alter Lab, which is basically the profile of early humoral signature of COVID-19 immunity. So let's start. The relentless spread and the unpredictable nature of disease caused by SARS-CoV-2 continues to paralyze the globe. If we look at the numbers, actually the numbers are from yesterday, there is more than 230 million confirmed cases of COVID-19, which leads to more than 4.7 million deaths. The introduction of potent vaccines against COVID-19 has inspired the hope that the end of the pandemic is in sight. So since December 2020, when the vaccine were introduced, there has been like enormous effort to vaccinate the population with currently emergency approved vaccines. Actually, one is already fully approved. We have 3 in US and 4 in Europe. However, the slow vaccine rollout, the vaccine hesitancy, and which was something which is even much more important, the emergence of new variants, the variants of concern, has led to the rapid and recurring increases in the cases of COVID-19. So actually right now we see the increase in cases, which is due to variant Delta, we don't know how many of these variants will happen and how they will develop. So there is still the urgent and continuous need to characterize the humoral antibody response, and it correlates with disease outcome. We need that to better define the biomarkers to support clinical care, as well as the therapeutic and vaccination strategy. There has been many different correlates of COVID-19 outcome described. I will start with one of the most important, the antibody. So both the neutralizing antibody as well as the antibody titer, so amount of antibody which is triggered by the virus, has been the correlate of COVID-19 outcome, but also the Fc receptor activity, the viral load, the altered B cells and T cells frequency, as well as the innate immune response are all the correlates of COVID-19 outcome. I don't think so I need to explain to all of you the entire cycle of COVID-19. However, briefly, the SARS-CoV-2 is spread droplet-wise. It enters our nose and mucus and then go lower to respiratory tract. The virus contacts with our cells through the spike protein, indicates the receptor binding domain, which is recognized by AC2 receptor. The virus replicates within the cells, and then it's recognized by antigen-presenting cells and trigger the immune system to produce the antibody. So the antibodies are currently the strongest correlate of immunity against the COVID-19. The antibodies have different function. They have actually much more than neutralization. I guess mostly you can hear about neutralizing antibody, how important they are in case of the both infection as well as vaccination. However, if we look at the picture of antibodies, we can distinguish 2 very important parts: the FAP, antigen binding fragment, and Fc, crystallizable fragment. So the FAP fragment, it's responsible for antigen recognition. So basically, we say that the FAP fragment is responsible for the neutralization and opsonization. However, I would say even more important is Fc fragment, which is responsible for the Fc-mediated effector function. For example, for phagocytosis, cellular cytotoxicity, for the granulation and cytokine release, for the neutrophil activation, such as netosis or ROS release. It also trigger the complement deposition, could lead to the epithelial barrier regulation or dysregulation, to B cell survival and regulation, as well as allergic reaction. And this Fc fragment is, and all the function dependent on Fc fragment are regulated by FcR binding, which are the FcR receptors are present on different innate immune cells. The Fc fragment is also the part when the protein are binding, for example, C1q protein from the complement cascade. And the regulation by itself is mostly due to glycosylation. You can see those end-linked glycans on the Fc part, and isotype, so the type of antibody by itself. In my lab, in Alter Lab, we mostly rely on the system, which is called system serology. So basically, it's a platform of different techniques, different assays, which allows us to characterize the immune profiles, the antibody profiles of the different, usually big cohorts of individuals. And it's a high throughput system, which leads to the very huge data set. So basically, the system serology can be divided into two different types of assay. First of all, there will be biophysical assay, which characterize mostly the character of the antigens, the epitope binding, the Fc receptor binding, as well as glycan on the Fc part. And the second type of the assays will be the functional assays, which characterize the functions of the antibody as Fc-mediated functions. So basically, we will see which cells can be activated by the antibodies and which specific functions can be triggered. So as I said, all assays will give us a huge data set, which needs to be analyzed by computational methods and different machine learning techniques, which helps us to understand and then define the profiles of humoral immune response. Let's start with biophysical assays. I will briefly explain the main techniques, the main assays which we are using, and which my results, my further slides will be mostly about. So the most important, or the main assay which we are using, is Luminex assay, which leads us to the isotyping and Fc receptor binding profiling. So basically, how does it work? So Luminex is a high throughput assay, which allows us to identify different antibody, different antibody of different specificities. So basically, we use the fluorescence beads, which have unique signature. So every bead is basically different color. And those beads are coupled with the antigens. In this case, in case of this work, they will be coupled with SARS-CoV-2 antigens. And then the antigen coupled beads are incubated with the serum sample, so with the antibody which we want to characterize. We will allow the formation of the immune complexes, so the antibody from serum will bind to the antigen on the surface of the beads. And then the last step will be the addition of the secondary antibodies, which have the fluorescence label on it. So we can add either the isotyping antibodies of FCR binding receptors, and then we can read the intensity of the signal. So here you can see again, how does it work? So we have beads with antigen coupled on it. We have antibody from the serum, and then the secondary antibodies, which are P labeled. So the readout will be based on this flow cytometry. So first we will record all events, so all beads which we have in our assay. Then we can distinguish different reagents, as I said, based on different fluorescence signature. So for example, we will have the reagent which has the RBD domain coupled on the surface, the other domain of the virus, as well as unspecific controls. So usually as a positive control, we will use flu antigens, because all of us has antigens against, has antibody against flu. And as a negative control, we will use Ebola antigens, kind of expecting that nobody will have the Ebola antibody in it. Then we read a fluorescence intensity. The MFI will be kind of equivalent to the amount of antigens which were in the serum samples. In case of this work, as I mentioned, we are using different SARS-CoV-2 antigens. So I want just briefly mention the antigens which we use in this work, which I will be referring later. So we are using a spike protein, which usually we call S. This is like a full spike. However, within full spike protein, we can also use the receptor binding domain, the RBD, as well as subunit 1 of the spike, which is called S1. So it will be kind of like the upper part of the protein, and subunit 2 of spike, S2, which will be kind of a lower part of the protein. We also use nucleocapsid N as one of the antigen, because this kind of tells us about the infection. And if we have samples from people who are vaccinated and infected, this is the antigen which basically distinguish both either infection and vaccination. What isotype and FCR do we measure? So we measure different antibody isotypes, such IgG, IgA, and IgM, and in IgG we can measure different subclasses as IgG1, IgG2, IgG3, and IgG4. For Fc receptor binding, we can measure the Fc gamma receptor 2A, which is activating receptor responsible mostly for phagocytosis, Fc gamma receptor 2B, which is the only inhibitory receptor, also Fc gamma receptor 3A, which is activating mostly on the cytotoxic NK cells, and 3B, which is mostly on neutrophils. I said mostly because they are, of course, different receptors on different cells, but this is like kind of the default, what we are called. Going back to system serology, I introduced the biophysical assays, but what about functional assays? Actually, in my work today, I would show the biophysical assays, mostly the Luminex. However, I think it's interesting and important to understand the entire system to better imagine how we can profile the humoral response. So within functional assays, we can distinguish 3 different assays: antibody-dependent phagocytosis, complement activation, and NK cells activation. Let's start with antibody-dependent phagocytosis. This is also beads-based assay. It's basically the same idea. However, in this case, we are using different beads. They are purely fluorescence beads and not magnetic beads, as in case of Luminex. We do the same. So we couple the antigen to the beads to have the kind of platform for that. Then we add the serum with the antibody. We form the immune complexes. And the next step will be adding the neutrophil or THP1 cells, which are phagocytic cells, and then allow the cells to engulf beads. And basically, we will measure the intensity of the beads inside the cells. So basically, whatever is eaten by the cells will give us the signal. The second assay, it's antibody-dependent complement deposition, ADCD. The same idea, we couple beads with antigen, we create the immune complexes, and then we add the complement components, in this case C3, which will bind to the C3 or C1q, both of them, which will bind to the antibody, and then we will add complement antibody, which will give us the measurement for the complement, the position of complement activation. And the last assay, it's antibody-dependent NK cell activation. This is not beads-based assay. This is like the plate assay, so kind of like ELISA. In well, every well of the plate we coat with the antigen. We add serum to form immune complexes, and then we add NK cells. And then we measure cytokine release, which is basically the final outcome. So now I will switch to the results section. So if you have any question, I'm happy to answer.
Thanks, Paulina. I think we don't have any questions yet, but I will let you know as soon as we do.
Okay, perfect. So let's start with results then. just to put all of us on the same side, I will briefly describe the cohort which we use in this study, the cohort to characterize the immune response or the early humoral profile of the COVID-19. So we characterized the individuals which basically showed up at the emergency department of MGH. The individuals were divided into two main groups, into survivors and non-survivors of COVID-19. And within survivors, we can distinguish individuals with moderate or severe disease. So the moderate individuals were hospitalized and they required the oxygen support, but they didn't need the mechanical ventilation. The severe individuals were intubated and they survived up to 28 days. And the non-survivors were people who basically died within 28 days since the admission to the hospital. So we analyzed the samples. We basically put them in kind of time beams. So we basically were interested in days of from symptom onset. So we had kind of 4 different time beams. The first one will be day 0 to day 3 from symptom onset. The second one will be from 3 to 6, then from 6 to 9, and from 9 to 12. So as I mentioned, we mostly focus on the characterization of the early humoral profile. That's why we focus up to day 12. So here you can see basically the first attempt to profile the antibody response in a cohort of acutely ill COVID-19 patients. So here you can see the different isotypes, the IgM, IgG1, IgG3, and IgA. This is the antibody response in 3 different groups. So in blue, you can see the moderate individuals, then in yellow, severe survivors, and in the red, non-survivors. So each row is the antigen. So we analyze RBD-specific antibodies, spike-specific antibodies, S1, S2, and nucleocapsid-specific antibodies. And then we can see the antibody response at different time points since symptom onset, so 0-3, then 0-6, 6-9, and 6-12. So as you can see, we actually notice in most of the groups the decrease of antibody titer in people who died within 28 days. So you can see that both IgM, IgG1, IgG3, RBD specific are decreased in their red group, so in people who died. We see similar trend for spike, S1, S2, and nucleocapsid antibody. Mostly this effect is visible for the early time points. So I would say at 9 to 12 days, we already kind of see that the level is more balanced than at the very beginning of the disease. And what we can actually claim here, that was our first observation, that this early evolution of the humoral response, the lower antibody titer, might be actually the signature of the COVID-19 mortality. But of course, we need to be careful with this kind of signature and correlation. So we further did much more detailed analysis to make sure that we can actually say that this is kind of a signature of the COVID-19 mortality. So I want to mention for you to remember that the antibody response, it's never the single antigen antibody response. It's always a polyclonal antibody response. It's a mixture of different antibodies in different amount and different affinity, so different strength, how they recognize the antigen. So we asked ourselves, how does it look like, not when we focus on the single antigen specificity, but when we take in account the entire polyclonal response. So basically here we wanted to see what's the relationship between different antibody specificity over time. So I know that this heat map it's a little bit complicated, so I will walk you through. So when you look in the columns, we have different time points. So first column will be time point 0.3, second 3.6, etc., etc. Then when you look at the rows, we have different disease severity. So the first row will be people with the moderate disease, then we will have people with the survival of severe disease and non-survivals. But when we look even more deeper into every square, the big square, you can see that it's a 5 by 5 square, big square containing 5 by 5 small squares, and every column here will be again the antigen. So it will be either spike-specific antibody, S1, S2, N, and RBD. And within those, we will have all isotypes. So we will have spike-specific IgM, G1, 2, etc., etc. But from the other side, we will have exactly the same description. So we will have different specificity and specific isotypes. So when we, for example, look at this small square marked in blue, this will show us the correlation, the Spearman correlation between S2 specific IgG3 antibody and RBD specific IgG3 antibody. So on this graph, you can see only the correlation of the relationship between different antibody response, which are significant. And from this heat map, at the first glance, as the first moment, we can already see that there is higher polyclonal coordination of the humoral immune response evolution, meaning like at the very first time point, we can already see that in COVID-19 survivors, mostly in the severe survivors on the day 0-3, this coordination is much bigger. You see much more orange color, which means that there's more relationship between those antibodies. But what does it actually mean? And is it actually influenced by the group size? Because as you remember, when I described the cohort, we have slightly different numbers of people in different groups. Of course, there is more people who had moderate to severe disease, and there is less people who died. So we wanted to make sure that whatever we see on this graph, it's actually a true thing. So we actually, one of them, colleague of ours, which is a computational expert, he did 2 different strategies to make sure that whatever results we see are true. So basically, first of all, he higher up the correlation coefficients value, and he only took in account the values which are larger than 0.6, so which are more significant. And the second of all, he did the downsampling strategy. So the machine learning basically choose and calculate the Spearman correlation for 500 randomly sample runs. And on this graph, you can see the numbers of correlation larger than 0.6 in this 500 randomly chosen groups. And we see very similar trends. So this is not dependent on the group size. This is kind of a real thing. Then in severe survivors, and also a little bit higher in moderate survivors, the correlation, the relationship between polyclonal antibody evolution, it's much higher. But then we wanted to know if this is only like a single point or time point related thing, or this is like a over time characteristic of the different disease severity. So we asked this question: could specific longitudinal humoral features could resolve the survivors from non-survivors? And to answer this question, we did, or Chansey did, another kind of complicated computational analysis. So basically, he used the pairs of nested linear, mixed linear models across each subset of subjects and measured the significant association between antibody levels and clinical outcome. So how to read this graph? What you can see here, we have the p-value from the likelihood ratio test. So the higher value will be more important. So everything in the higher part will be kind of more significant. and then t-value will basically show us the clinical outcome. So either more to the left, we will, for example, on the first part, we will see this relationship in the group of people who did not survive the COVID-19. And in this case, on the right-hand side, we will see the correlation, which are important in a group of people who had the severe COVID-19. And when we look at the results, we basically notice that the S2, so spike domain 2 specific response, are selectively reached in survivors of COVID-19. So when we take a look at the first graph, you see that S2 FCR2b antibody able to bind to receptor 2b or 3a are enriched in survivors. And when we compare against survivors and non-survivors and survivors, we see mostly the S2 specific antibody features. So, for example, 2B binding to receptor 2B, 3B, 2A. We can also see some of S1 and RBD, but the first thing which you can actually notice are the prevalence of the S2 specific response. The next aspect of this entire analysis was to kind of understand if this is like only SARS-CoV-2 specific response, because what we can see when we check the ratio of the IgM, which are the antibodies produced first in the moment of actually first encounter with the virus, and IgG, which are produced more in the second encounter or later during the infection, we see almost simultaneous evolution of those two antibodies, which kind of lead us to the conclusion that it's either the very rapid maturation, because we see a lot of IgG at the same time point, or it's maybe the potential expansion of the pre-existing cross-coronavirus immunity. So there's quite a few different coronaviruses circulating amount, which lead to the common cold, like kind of flu-like symptoms and flu-like disease. Excuse me. And we can distinguish alpha and beta coronaviruses. So we have 229E, which is alpha coronavirus, NL63, which is alpha. We have OC43, which is beta coronavirus, HKU, beta coronaviruses. So they are all common human coronaviruses. And we also have a group of other, which are more dangerous and not as popular as the common. So we have MERS and SARS, which you all heard about. And actually, OC43, so the one here shown in blue, so the beta coronavirus, it's the most common coronavirus in the population of United States. So we ask ourselves, what's the influence or what's the relationship between the antibody response to SARS-CoV-2 and the antibody, like the existence of antibody response to circulating common beta coronaviruses? So there is something called the antigenic scene. This is a phenomenon firstly described for the influenza, which basically shows that pre-existing immunity to particular strain of influenza prevents the boosting of orthogonal strains. So basically, if we got infected with virus A, and then we got infected with another variant of the virus A, we have a little bit more response to the virus A, and then slightly less response to the original virus. And if we have, for example, the contact with another type of the virus, so we, again, will have less of this new virus, but more of the response to the first virus. So basically, the contact with another virus will increase the response to the original. And we were kind of afraid that maybe this, whatever we see, it's influenced or it's biased by the OC43, the common cold virus, because all of us had a contact with that. So maybe whatever we see, it's not really from SARS-CoV-2, but it's also the OC43 response. So what we actually checked was the OC43 antibody profile. And what we notice is the higher OC43 RBD specific IgM as well as IgG level in in survivors. So the non-survivors will have decreased level of the OC43 specific antibody in line with the SARS-CoV-2 response. So we also investigate if this is only the time point related or this is also the same happening over the time. So we check the OC43 RBD specific humoral immune profiles, why they integrated into the pair-nested mixed linear models. And what we observed, let's first focus on the graph on the left-hand side. We noticed that the spike-specific RBD and S2 IgG1 are again popping out, but interestingly, we also see OC43 antibody response, which is like much more higher or much more important in the survivors. So the importance of the early expanded OC43 IgG level as well as the FC binding kind of lead us to the observation that this might be correlated or important to the disease trajectory. When we compare the survivors, but severe and non-severe, again, OC43 pop up, and we can see that it's enriched in survivors with the severe disease compared to moderate, which might potentially be important as a mature expanded OC43 response in the resolution of the disease. And when we compare the non-survivors with mild survivors, we see the RBD IgM levels, which was the most discriminatory features. And it was kind of like a top predictor for the severe survivors compared to non-survivors, selectively expanded. So then we dig more into the OC43 SARS-CoV-2 immune response, and we check the correlation between the SARS-CoV-2 antibody response. So on the x-axis, you will see the spike S1, S2, and an RBD different isotypes. Again, then the correlation against the OC43, this case RBD different isotypes IgM, G1, G3A, and also FcR receptor binding. So again, we can notice that there is the highest correlation or the highest relationship of the OC43 SARS-CoV-2 immune response in people who survive but had the severe disease. And we did again the same strategy. So we did downsampling to make sure that whatever we see, it's not the effect of the sample size, but it's a real thing. And we see only the positive correlation between OC43 and SARS-CoV-2 immune response, which would indicate that the pre-existing common coronavirus immune response is damped or blocked during the evolution. And the positive correlation, which we mostly see, suggests that the pre-existing common coronavirus immune response give rise to evolving SARS-CoV-2 immunity. So we ask ourselves, is it only a characteristic or is it only specific for acute COVID-19? Because so far we analyze only people who are severely sick, or maybe the same thing happened or the same thing phenomenon it's observed in asymptomatic COVID-19. So to answer this question, we analyzed a cohort of community-acquired mild or asymptomatic COVID-19 individuals. So we got the samples from people who basically acquired the SARS-CoV-2 virus from a community. They don't even know that they are sick because they have had either completely no symptoms or very mild symptoms. So those people basically were regularly checked. There are employees of SpaceX. So they were checked regularly and they did the survey if they had any kind of COVID-related symptoms. So we characterized those people or divided into different levels, level 0, 1, and 2, based on severity of their symptoms. So we basically gave 2 points to every symptoms which is really related to SARS-CoV-2, such as loss of smell or taste, higher fever or fever, chills or cough, or 1 point to the more to the weaker symptoms or the symptoms which might be not directly connected to COVID-19, but still might lead to, so such as fatigue, headache, sore throat, or muscle ache. We summarize all those symptoms, and the score of the points basically help us to put those people even to level 0, which is no symptoms, level 1, which is mild or very mild symptoms, and then level 2, which is moderate, but please still keep in mind that this is not the same moderate group as from the first cohort, because those people are not hospitalized. They just had kind of the symptoms which might lead to the conclusion that they got COVID-19. So then we did the system serology on these samples, and we check what's the difference between this antibody response at the time point pre-infection and post-infection, because we were also lucky to have the serum samples before those people are actually feeling sick and kind of got COVID.
Paulina, I've just noticed that there's a question here from Bob Hansaker, which I will voice now, if that's okay.
Yes, sure.
The question is this: I may have missed this, but when you studied the moderately to severely ill patients, were there differences in age across the groups? And if so, how did you control for age?
Yeah, so of course there were differences in age, but we check if the age, so age was also included in the model, and we check if age has any kind of influence of the the antibody response or the antibody profile, and we excluded that. So we know that age is not responsible for, at least in our study, in the group of people which we analyzed, age is not responsible for different antibody outcome. So as you remember, in the mixed linear model was basically controlled on based on different demographic information such as age, gender, BMI, etc., etc. So we basically controlled and excluded all different bias based on different demographic information such as age.
Got it. Thank you very much.
Sure. Okay, so coming back to our asymptomatic or very mild individuals, we characterize the antibody response of those people pre and post-infection to see if there's any differences, especially in OC43 immunity, because we know that there is some effect of this antibody response, and after infection. So whenever we, so the post-infection time point, it's basically whenever those people reported that they have symptoms which are COVID-related, we kind of took those antibody profile as a post-infection time point. So first of all, you can see that there is no differences observed in OC43 specific antibody profile across individuals. So there is nothing really specific what we see if we compare the antibody, not between the time points, but just the level. However, after infection, we can see that OC43 specific IgG were selectively enriched in individuals that remained asymptomatic or have single or mild symptoms. So as you can see for IgG1, there's significant, very actually visible increase in OC43 antibody response before and after infection. So then we also ask if there is like any SARS-CoV-2 S2 specific IgG humoral response. Why S2? Because S2 is very conserved part of the spike. So basically this will be the part or the antibody which will be very similar across different types of coronaviruses. And what we can see, why the S2 specific IgG humoral immune response were expanded across all individuals, the antibodies which were able to bind to FcR2a and 3a receptors, which are phagocytic and cytotoxic receptors, were expanded or were elicited in asymptomatic individuals. So you can see that here we see very low level, and here we see for the people who are who having the higher number of higher points of symptoms are the highest level of the antibody, which SARS-CoV-2 antibodies, which are basically able to bind to FcR receptors. And the expansion of this S2 specific response was derived from pre-existing common coronavirus immunity and might contribute to the disease severity via Fc-mediated antiviral control and clearance, because those antibodies able to bind to Fc receptors might trigger the phagocytosis and the cytotoxicity. So to briefly summarize all those observations, I hope I was able to convince you that class switch SARS-CoV-2 S2 specific humoral immune response evolved rapidly. So we see it at the very early time point since symptom onset, and it selectively expand in acute survivors of COVID-19. We can also see, based on our results, that IgM and IgG response to the common beta coronaviruses such as OC43 were significantly expanded in survivors who experienced milder disease. We can also observe that the earliest OC43 specific response were linked to the acutely evolving SARS-CoV-2 response across the disease spectrum, which points to the importance of the leveraging pre-existing common coronavirus immunity to control and then ultimately clear the infection across the disease spectrum. And what is the most important, or at least I think it's very important, that when we don't see the original sin, we actually see that pre-existing cross-coronavirus immunity may accelerate the evolution of cross-reactive humoral response to the highly conserved region of the virus, which may point to the critical target of COVID-19 immunity. And at the end, I would like to acknowledge multiple people which help with this study. So first of all, my PI Galit Alter, who provide the samples and equipment and then the entire everything to work in the lab, and also to Chanzi, which is a computational person from MIT and who helped to analyze and understand all of this huge data set, as well as multiple collaborators which both give us samples and antigen to use for the bits coupling, and people who help with small assays and discussion and everything to make this study as it is now. And thank you very much to all of you for your attention, and I'm happy to answer all questions.
Thank you so much, Paulina. That was really wonderful and thought-provoking. I don't see any additional questions in the Q&A box right now, so I will remind the audience to please post any that you have there. And I will take this opportunity to jump in with a question myself that kind of follows up on the earlier question about age. It's tempting to think, given the ultimately seemingly protective value of cross-reactivity having to do with previous exposures in terms of long-term outcomes, that it might be protective to have had an earlier exposure, and that might be sort of a thing to hope one has. In terms of age, I might naively at least think that older people would have had longer cumulative opportunities for exposure that could ultimately prove protective through cross-reactivity. And I'm wondering then, how you explain sort of the what is observed as the greater risk in that age group. Is it simply that that age itself is a risk factor for severe outcome independent of these exposures, do you think, or how do those things interact?
Yeah, absolutely. So you're fully right that younger people will have less exposure to these common coronaviruses. So of course, there will be some kind of a correlation between how old and how much exposure do we have to OC43 or other coronaviruses. And there is like many studies trying to show what's the difference between younger age and between them, how much contact do we have with that. However, like what is important for us, and like the problem is that there's many different contradictory studies. So some people are trying to show that those response or show that this response to different coronaviruses, it's actually not helpful and might be not helpful, like, or, you know, in the younger group of people who don't have so much, maybe we don't see this effect. However, in the people who are in the age, or maybe also this both cohort were from US, where OC43 and other coronaviruses are more prevalent, there's definitely the influence and like correlation, and there is no antigenic scene, so there is no bad effect from contact with this coronaviruses.
Thank you very much. Another question has just popped up in the Q&A box. It's from Bill Crowley, who says, excellent talk. Given that commercial monoclonal antibodies have been found to be therapeutic in COVID, have you been able to epitopically map them and see which of your panel of antigens overlap with them to guide further development? So
unfortunately, we haven't been able to do it. But I know that many people is actually trying to find what's the most important epitope for the monoclonal antibody therapy. And actually, as I mentioned, we saw many of these S2 specific antibodies, which has been shown and proven that many monoclonal, like lately in the hamster model, they show that they are protective. So might be that we should focus more excuse me, focus more and try to understand what's this S2 conserved domain, specific antibodies, what's their profile, how they work. But it looks like there is something really related to this common cross-reactive antibody.
Thank you. And then there's just one more question in the Q&A box, which has just been answered by the person who raised it, which is, can we post a link to the paper? So for anyone who's looking to read more about Paulina's work, thank you, Bob, for posting that link in the Q&A. And I think that ends the questions, and we're about out of time as well. So I will just take this opportunity again to thank you, Paulina, so much for this talk on some really fascinating and obviously current and really important work. And we will look forward to hearing where it goes as you continue working. So thank you so much, Paulina, and thanks to everyone for attending.